Good morning, students. Today we are going to deal with the last part of your biological classification, the most sought after kingdom, that is kingdom Animalia. This kingdom has been studied or researched on exclusively and is one of the largest kingdom belonging to your biological system. So let's start with kingdom Animalia, of which you and me are a part of. Before we move to the classification part, we will discuss the general characters of kingdom Animalia. Now, like all of you know, all organisms belonging to animal kingdom are eukaryotic in nature and they are multicellular in nature. The cells of these organisms does not possess cell wall, and plastids and vacuoles are also absent. Since plastids are absent, they cannot synthesize chlorophyll and thus are not photosynthetic in nature. Since they cannot prepare their own food, these organisms rely on other organisms for food, which makes them heterotrophic in nature. They store this accumulated food in the form of glycogen in their body. Now, animals have certain special characteristic features which you will not find in the previous kingdoms. One of them being that they have sense organs. Sense organs are special organs present on the body of the organism which help the organism to be aware to his surroundings and be more receptive. The second one, they have locomotory organs. Locomotory organs are organs which help the organism or the animal to move about freely. This you will not see in plants. Now, for proper functioning of these locomotory organs, the body of the animals comprises of muscles. And for enhanced receptiveness of these muscles, there is nervous coordination. These are all terms that you will be studying in detail further on, but I am just introducing them to you now. Moving on, these animals have evolved consciousness which gives them the capability to change according to change in the environment. Reproduction in kingdom Animalia is generally sexual in nature. However, some lower animals which were poorly evolved show asexual reproduction. Now that we are done with the characteristic features, let's see how animals are classified into various groups or phyla. So under the heading criteria for classification, we will be looking at how they are classified into different phyla. Since this kingdom comprises of varied living organisms, we are going to see on what basis they are classified into different phyla. So under the heading basis of classification or criteria for classification, we will be studying 7 different criteria due to which the organism may be grouped into one of the phyla. The first criteria is levels of organization. When you are classifying, there are 5 levels of organization. The first is cellular grade of organization, in which the body of the organism comprises of a mass of cells which are performing different functions. These cells are all individual in nature and it is not grouped into tissues. A very common example of animals showing this grade of organization are sponges. The second one, you have tissue grade organization, wherein cells come together to form specific tissues, and these tissues in turn will carry out different functions like absorption, respiration, excretion, etc. A common example showing tissue grade of organization is Hydra. The third one, you have organ grade organization. So, as organisms evolved, tissues formed groups called organs. Each organ performed a specific function inside the body. So organisms showing organ grade organization, you can give an example as flatworms. The fourth one and the most evolved level of organization is organ system grade of organization, which is commonly seen in humans. Now, like you have a digestive system, a nervous system, an excretory system. This system comprises of various organs. So, here you have organ system grade of organization. So, this commences the first criteria for classification, that is levels of organization. The second criteria you have according to body plan. In animals showing cellular grade of organization, the entire body is covered by minute pores which is responsible for exchange of gases and other materials. So you do not have specific tubes inside the organism or the animal. Such a plan is known as cell aggregate plan. The second one is the blind sac plan, in which the body of the animal generally has tissue grade organization and it is comprising of one single opening which acts as an inlet as well as an outlet. So ingestion as well as egestion is taking place through the same opening. The third one is tube within tube plan. This plan is generally seen in organisms or animals which are showing organ or organ system grade of organization. In these animals, you will find an inner tube with two openings, one at the top and one at the end. So an anterior opening and a posterior opening. The anterior opening acts as a mouth and the posterior opening acts as the anus. With this, you have a tube within the tube plan. So the body of the organism comprises of an inner tube surrounded by a body cavity, okay, and there are two openings, anterior and posterior. With this, we finish the second criteria, that is body plan. The third criteria is germ layer formation. Now, during embryonic development in higher organisms, that is organisms or animals showing tissue grade organization, during the embryonic development, the male and the female gamete fuse to form zygote. This process is known as fertilization. Haploid zygote now starts undergoing multiple divisions known as cleavage. So one single zygote cell starts dividing to form 2 cells, 4 cells, 8 cell stage. This 8 cell stage is known as morula. The 8 cell stage comprises of 8 cells which are aggregated to form one ball-like mass. After this, the divisions which follow start creating a cavity in the center of the morula. This process is known as blastulation. So conversion of an 8-celled morula into a 16-celled, 32-celled blastula with a central cavity known as the blastocele is known as blastulation. The cells of the blastula start undergoing a further process known as gastrulation, wherein the outer lining of the cells start moving towards the upper portion. This process is known as convergence. They start converging towards one single point. From one point, they start moving inside the cavity. This process is known as invagination. Now these cells which have moved inside or invaginated inside the blastocele start moving away from each other. This process is known as divergence. And eventually they start convoluting or joining back again, forming a cavity. This process is known as gastrulation, and at the end of this process, the structure that is formed is known as a gastrula. Now, this gastrula may consist of 2 or 3 layers. These layers are called germ layers. Depending on evolution, the animal may either have 2 germ layers or it may have 3 germ layers. The outermost germ layer is known as the ectoderm. The middle layer is known as the mesoderm, and the innermost layer is known as the endoderm. So if an animal has 3 germ layers, that is the ectoderm, mesoderm and endoderm, it is known as a triploblastic animal. And if an animal has 2 germ layers, just the ectoderm and the endoderm, mesoderm is absent, then that animal is known as diploblastic. So the third criteria, which says segregation of animals based on germ layers, you may either have triploblastic animals or diploblastic animals. Moving to the fourth criteria, that is symmetry, applies to when you cut an organism into 2 parts, whether those 2 parts are equal or not. Okay, based on that, you have 3 types of symmetry in animals. Organisms which cannot be divided into equal parts are called asymmetric in nature. If you can divide an organism by cutting it through the center into 2 equal parts, this type of symmetry is known as bilateral symmetry. The third type of symmetry is wherein you can take a cut on that organism through any plane and divide it into equal parts. Such an organism shows radial symmetry. This is the fourth criteria for classification. The next one, presence of body cavity. Lower organisms were flat in nature, so they did not possess any body cavity inside. Such organisms or such animals were called acoelomates. Example, your flatworms. Next, as evolution started taking place, certain organisms became long, slender and cylindrical in nature with presence of a body cavity. But this body cavity was not very distinct and it merged with the body wall. Such a type of organism was said to be pseudocoelomate. Example, you have your roundworms. Now, in the third category, you have organisms which are highly developed, showing an inner tube-like structure or the digestive tract, surrounded by a proper distinct body cavity, enclosed by a layer called peritoneum. Okay, the cavity is known as coelom. The wall of the coelom is known as peritoneum. These are words you will be hearing very frequently later on, so I am getting you used to them. And after the peritoneum, you have the body wall. So with such distinct coelom, the organism or the animal was said to be eucoelomate. We are all eucoelomate in nature. So this is the fifth criteria for classification based on coelom. The sixth one, segmentation. Organisms which can be divided into equal segments are called segmented organisms. Organisms which cannot be divided into equal segments are called unsegmented organisms. Animals belonging to unsegmented category are generally also pseudocoelomate in nature. In segmented animals, they show distinct equal distinctions or equal segments, which you call metamerism. The last criteria for classification is presence of a notochord. Now, notochord is a hard dorsal solid structure which is present within the body of the organism, dorsally in maximum cases, ventrally in a few cases. Okay, and this acts as a support for the other organs present inside the body. So based on presence and absence of notochord, you have 2 types. If notochord is absent, the animal goes under the category of non-chordates, and if notochord is present, the animal goes under the category of chordates. In chordates, the more evolved animals have replaced the notochord with a sturdier and more flexible structure known as the vertebral column. Such animals are called vertebrates. With this, we complete the 7 criteria for classification. So let's revise again. First one, you have levels of organization comprising of cellular grade, tissue grade, organ grade, organ system grade. The second criteria you have based on body plan. You have 3 plans: cell aggregate plan, blind sac plan, and tube within tube plan. Third criteria for classification based on the type of germ layers present. You have diploblastic animals and you have triploblastic animals. Fourth criteria for classification based on symmetry. You have asymmetric animals, animals showing bilateral symmetry, animals showing radial symmetry. Fifth criteria for classification based on presence of coelom or body cavity. Absence of body cavity, acoelomates. Presence of body cavity which is not distinct in nature, pseudocoelomate. They have false coelom present. And presence of a distinct body cavity or coelom, you have eucoelomates. Sixth criteria for classification based on segmentation. If the body of the animal is segmented, then they come under segmented category. If there are no distinct divisions on the body, they are unsegmented animals. And the last one, presence or absence of notochord. If notochord is absent, they are non-chordates. If notochord is present, they are chordates. With this, we come to an end of this video. Thank you.